Okay, so in section 2, we're going to talk a little bit about how viruses are classified. I've already mentioned a little bit of this previously, but we'll talk about it in more detail now. So when it comes to classifying viruses, we can't use that same system that we use for bacteria, plants, or animals because, again, viruses aren't cellular. They don't have the organelles or ribosomes or their own metabolism. So virologists are going to classify viruses using different characteristics, and that's really going to be characteristics describing their structure, their replication strategy, or potentially how they cause disease. So one of the ways it can be classified, again, is by the type of nucleic acid. I already mentioned that it could be DNA or RNA, and that it could be single-stranded or double-stranded nucleic acid. So this is one of the most important distinctions because it's going to directly affect how the virus replicates once it's actually inside of a host cell. So for example, a double-stranded DNA virus is often just going to use the host's own enzymes in the nucleus in order to replicate. But if we're talking about an RNA virus, that's typically going to have to replicate in the cytoplasm, and it's going to have to bring with it and use its own RNA enzymes because we don't have those. Other things, the shape and the structure, the capsid that we just talked about. So the protein shell could take on 3 different shapes: helical, icosahedral, or complex. And this is going to affect how the virus attaches to the host cells and how stable it is in the environment, depending on whether or not it has an envelope. Also, we can describe viruses in regards to their host range, so the types of organisms that this particular virus can infect. So some viruses are the specialists, so they only infect one species or even one type of cell within that species, while others have a broader range of hosts that they can infect. And so the example I gave previously is that HIV can infect human immune cells, while influenza A can affect a broad range. So pigs, us, and birds. So the last thing I want to talk about is how they're grouped according to what's referred to as the Baltimore classification. So this is based on how they make their mRNA. And this system is going to divide viruses into 7 different classes depending on their nucleic acid type and their replication method. So for example, class 1 viruses, these are going to be our double-stranded DNA viruses. And then class 4 would be something like our positive sense RNA viruses. Class 6 is our retroviruses. So we'll talk about these later on in this classification system. So these are just tables giving you examples of the family and the representative genre. I don't really want you to know all of them, but there's a couple I do want to point out, and these would be the ones that I would focus on. So this slide is showing you DNA viruses. So remember, DNA viruses tend to have more stable genomes than RNA viruses. So that means that it's going to have slower mutation rates and a little bit more predictable disease patterns. So a lot of these viruses are going to replicate in the nucleus because they're DNA viruses, meaning that they can establish really long-lasting or latent infections. So a really good example is variola virus. So this is a member of the Poxviridae family, and it's the causative agent of smallpox. So variola has a linear double-stranded DNA genome, and it is unique among DNA viruses because it replicates entirely in the cytoplasm. So remember, I just mentioned that usually they replicate in the nucleus. So historically, smallpox was one of the deadliest infectious diseases, but also the first to be eradicated through mass vaccination campaigns. So this occurred around 1980s-ish. Another important DNA virus is the herpes viridae virus. So this is going to include herpes simplex virus 1 and herpes simplex virus 2. And these viruses also have a linear double-stranded DNA genome, and they are going to be transmitted through direct contact with mucous membranes or some sort of lesion. So HSV1 is typically going to cause oral lesions, whereas HSV2 is more commonly associated with genital infections, although there is some overlap. So a really classic mechanism of herpes viruses is their ability to establish latency in sensory neurons. So this can lead to, in periods of like high stress or something like that, it can lead to their reactivation, potentially, not always, and it could happen multiple times. So shingles is a good example of a virus that goes latent and then it comes back in light. Okay, another major DNA virus of clinical importance would be human papillomavirus, or HPV. So HPV has a circular double-stranded DNA genome and is transmitted through usually skin to skin and sexual contact. So not one or the other, both would be required. A lot of infections are really asymptomatic, meaning that our immune system can clear them readily. However, they can potentially be persistent, and there are some strains that are more high risk than other strains. So HPV 16 and HPV 18, those are the strains that can lead to cervical cancers or even oropharyngeal cancers, something like that. So what's really great is now that we have HPV vaccines, and this has significantly reduced the prevalence of the HPV 16 and HPV 18, as long as we're maintaining a good number of vaccinations in certain populations. Okay, this slide is talking about RNA viruses, and again, just kind of the ones that I point out here are the ones that I want you to know the details of. So unlike our DNA viruses, our RNA viruses typically have to replicate in the cytoplasm, and they're going to rely on their own RNA-dependent RNA polymerase. And as I mentioned, I think in the beginning, the viral RNA-dependent RNA polymerase lacks proofreading ability. And because it can't proofread, we're going to get high numbers of mutation rates in RNA viruses. It's not always a bad thing as far as the virus is concerned, because it's going to lead to really fast-paced evolution. And downside for us, it leads to an increase in the emergence of new variants. So one really important RNA virus is poliovirus. This is a non-envelope, positive sense, single-stranded RNA virus. It's transmitted primarily through the fecal-oral route. So poliovirus is going to affect motor neurons, and when it does, it can cause poliomyelitis. This can potentially be a paralytic disease. It's been nearly eradicated through global vaccination efforts, but they're actually having issues in Africa where the vaccine has actually reverted back to the wild type. So they're dealing with basically a vaccine-based endemic. It's localized in specific areas, not in the U.S. I actually did a presentation on polio a couple years ago at UF. Okay, another closely related virus in the same family is going to be hepatitis A virus. This also spreads through the fecal-oral route. It causes acute hepatitis. So that's not like hepatitis B and hepatitis C, because those aren't chronic, whereas hepatitis A is a chronic condition. So it is environmentally stable, and it can persist on surfaces for weeks and even months. So it really makes sanitation and vaccination the most important control method when you're talking about hepatitis A. Another highly contagious RNA virus would be norovirus. You may have heard about this in the news. Like, I just watched something on the news like 2 weeks ago about it on cruise ships. So it's really the leading cause of viral gastroenteritis worldwide, and it's transmitted through contaminated food, water, or surfaces. So it spreads very, very quickly. If there is an outbreak, it's usually going to occur in some sort of closed environment like a cruise ship or a dorm at a college or even nursing homes. So it requires a very low inoculation or a low amount in order to be infectious. So it can spread very, very quickly. And when it does, it's going to cause vomiting, diarrhea, and as a result of that, dehydration. But the good news is the symptoms are usually short-lived. Okay, RNA viruses. Continued. On this slide, so some RNA viruses are enveloped, and again, that's going to give them some sort of additional mechanism for host cell entry and as well as immunization, but also make them less stable in the environment. So a good example is influenza A. It's an envelope segmented negative sense RNA virus, and it's going to be transmitted through respiratory droplets. However, as far as its genome is concerned, the segmentation is really important. So the segmented genome allows for genetic reassortment. So this is going to cause them to kind of rearrange the genes that it has, or during viral replication, exchange genes between different host cells. So this is what really the segmented genome is going to be the major cause of antigenic shifts and the pandemics that we have, such as the H1N1 pandemic, things like that. So frequent antigenic drift is also going to explain why flu vaccines have to be updated annually. So why every December are you encouraged to get a flu vaccine if you had one 365 days ago? Well, that's because they have this segmented genome that's constantly undergoing genetic reassortment. So there's like 100% chance that the vaccine that you got for the flu last year isn't going to be effective against any of the genomes of influenza A that are circulating this year. So another globally significant enveloped RNA virus would be SARS-CoV-2. So this is a positive sense RNA virus, and again, it emerged in late 2019. And this is what caused the COVID-19 pandemic. So how it enters cells is through interactions between its spike protein and the ACE2 receptor. It has a really large RNA genome that encodes a lot of proteins that are really there just to help enhance replication and efficiency and immune evasion. Another one I want to point out is the rabies virus. So rabies is a negative sense enveloped RNA virus, and it's transmitted through the saliva of infected animals. So typically it's going to occur via bites. It is a neurotropic virus, so that means that it's going to travel along peripheral nerves to the central nervous system. And usually once it's there, you have 100% fatality once you start to see those symptoms. However, it is preventable through post-exposure vaccination, but this has to be administered very quickly before the symptoms are actually